Good morning. In this session, I would like to speak about transgenic animals. Dear students, before going to the concerned topic, I would like to ask a simple question to you people. What are transgenic animals? See, transgenic animals are the animals where their DNA is modified, means a foreign gene is inserted into the genome of the target animal. They are known as transgenic animals. The transgenic animals are genetically engineered and are also known as genetically modified organisms. The first genetically modified organism was engineered in the year 1980. So, which is the first transgenic animal? So, that is the first transgenic animal modified organism is the mice, transgenic mice, whereas Dolly, which was modified in the year 1996. So we will see the examples one by one. We'll take two examples in this session. The first one is transgenic cows. Transgenic cows are genetically modified cows, means GM cows. They have an extra gene or genes inserted in their DNA. These are the techniques or methods which are involved in the making of the transgenic cow. Transgenic cows is a multi-step process which involves 7 steps. The first one is designing the gene construct, second one is sourcing the transgene, making the gene construct, and fourth one is transfecting bovine cells. Fifth step is selecting for transgene positive cells. And sixth one is making a transgenic embryo using nuclear transfer. And the seventh one is the last step, which is commonly called as confirming the cow is transgenic or not. Okay, so these are the 7 steps which are involved in the process of producing transgenic cows. So hence it is commonly called a 7-step process. Now coming to the second example, which is transgenic fish. So like transgenic cows, transgenic fish were developed using a variety of transgenes methods, and the species since the first transgenic fish were produced in China in the year 1985. Since that time, transgenic gene knockout and gene editing technologies have been adapted for fish experimentation. These are the methods which are used to produce the transgenic fish. They are microinjection method, electroporation method, sperm mediated gene transfer method. So that is SMGT, sperm mediated gene transfer method. And the last one is retroviral infection method. We will see one by one. The first step, that is microinjection method. In this method, and this method is usually successfully used to produce the transgenic fish, and it is commonly used technique due to its simplicity and reliability. The use of the microinjection method results in higher survival rates for manipulated fish embryos than the electroporation method. Means the microinjection method is highly effective and survival rate of the target organisms are more, and hence it is the most established method for gene transfer in fish The second one is electroporation method. So in this method, the gene transfer in fish is effective, and electroporation has been preferred in many laboratories for gene transfer because of its efficiency, speed and simplicity. Hence this method is also very popular in the production of transgenic fishes. Third one is a sperm mediated gene transfer method. So that is SMGT. SMGT is a simple technique or a transgenic technique where the transfers of genes based on the ability of sperm cells to spontaneously bind to exogenous DNA and transport it into an oocyte during fertilization to produce genetically modified animals. The fourth one is retroviral infection method. This is a successfully used method with the retroviral vector to produce transgenic crayfish and a top minnows, and which is used as a tool for transducing sea urchin embryos. This is a valuable gene transfer technique for fishes. In general, the viral sequences into food fish may not be accepted by public, but it could be used for ornamental fish development. And these are the four important the techniques or methods which are used to produce transgenic fishes. What are the advantages of transgenic fish? So here there are many advantages. Among them, a few are very important. They are as follows. The first one is the growth rate of transgenic fish can be increased by 400% to 600%, and it is also used as a bioreactors for large-scale production of rare human therapeutic proteins or novel foods for specific dietary requirements. And the transgenic lines of tilapia. You know tilapia, it is the commonly jalebi fish. The transgenic lines of tilapia engineered to produce a human clotting factor 7, which is used in liver transplants and treating injuries. And this method is also useful to development of transgenic animals models, represents a revolutionary advance in the study of variety of diseases process. Means transgenic fishes are also useful to study the varieties of disease processes. Hence these are the few important advantages of transgenic fish. So dear students, in this session we have studied about transgenic animals by taking an example of transgenic cow and transgenic fish. I hope you all understood well. Thank you.